Welcome to this lecture on protein structure analysis, a university-level overview that guides you through modern techniques and their impact on biology and medicine, from experimental imaging to computational prediction, setting the stage for why understanding structure matters for function and drug discovery. This slide outlines the journey we'll take in this lecture, covering fundamentals, key experimental methods, computational breakthroughs like AlphaFold, practical workflows, case studies, and ethical considerations, so you get a roadmap of topics that build from basic concepts to applied research. In this introduction, we highlight why protein structure analysis is foundational, explaining how structural insights reveal molecular mechanisms and guide targeted therapeutics, and we briefly note primary techniques like X-ray crystallography, NMR, and cryo-EM, with the image showing typical lab workflows and cryo-EM reconstructions for context. Here we break down the four hierarchical levels of protein structure: the linear primary sequence, local secondary motifs, such as helices and sheets, the folded three-dimensional tertiary arrangement, and the assembly into quaternary complexes, with the illustration helping visualize how local folding leads to complex 3D forms. Protein structures are held together by several key interactions: hydrophobic packing drives folding, hydrogen bonds stabilize local motifs, salt bridges add electrostatic stability, and disulfide bonds create covalent links in certain proteins, all working together to maintain the native conformation under physiological conditions. Quaternary structure describes how multiple polypeptide chains assemble into functional complexes, exemplified by hemoglobin's alpha and beta subunits and viral capsids that package genomes, and these subunit interactions create biological units essential for functions like oxygen transport and viral protection. Traditional proteomics and structural methods leave gaps, such as an unseen proteome where many peptides remain uncharacterized, and a heavy dependence on sequence databases, while AI-powered tools are changing the field by enabling de novo identifications and expanding what we can infer from sequence and noisy data. AlphaFold represents a revolution in 3D prediction by using deep learning to convert genetic sequences into highly accurate structural models, which has dramatically accelerated research by providing precise starting points for experiments and informing drug discovery and biological hypothesis testing. Cryo-electron microscopy relies on flash-freezing biological macromolecules in vitreous ice, imaging thousands of particle projections, and computationally reconstructing 3D maps, which excels for large or flexible complexes and has opened access to structures that resist crystallization. The Tokyo University CHR-Aminte case highlights a high-resolution cryo-EM breakthrough, achieving around 2.02 angstroms, using Fab fragments for orientation control, overcoming membrane protein challenges, and demonstrating implications for structure-guided drug design against difficult targets. Sample preparation is critical. Protein purity and careful crystallization, or vitrification, determine data quality. And the photo of a scientist at work reminds us that meticulous lab technique and optimized buffer conditions are often the difference between success and noisy, unusable data. Data collection in structural biology requires precision, with X-ray diffraction demanding high-quality crystals, and cryo-EM needing careful sample preparation and massive image datasets. Both approaches have distinct technical hurdles, but together cover a wide range of structural problems. Modern structural analysis depends on computational tools like ReLIAN for single-particle cryo-EM refinement, the advancements in ReLIAN 4 for improved workflows, and a suite of complementary software that refines models and automates repetitive steps to achieve high-resolution results. The Protein Databank remains the central archive for atomic structures, while the Electron Microscopy Databank stores 3D maps. Visualization and validation tools such as PyMol, Chimera, DSSP, and ProCheck are indispensable for interpreting and verifying structural models. Membrane proteins pose special challenges because their hydrophobic surfaces resist crystallization and destabilize in aqueous solutions. So strategies like detergent screening, lipid cubic phase crystallization, and engineered fusion tags are often used to improve stability and structure determination. Antibody complexes are powerful tools for structural work. Fab fragments can control particle orientation to enhance resolution, and fluorescence-based size exclusion screening helps ensure only correctly assembled, stable complexes are advanced to structural analysis, improving final map quality. Data processing workflows turn raw images into refined structures through robust pipelines that handle enormous datasets, with critical steps including data cleaning, noise reduction, particle picking, classification, and iterative refinement to reach reliable, high-resolution reconstructions. Validating protein structures relies on quantitative metrics like Ramachandran plots, clash scores, and Molprobity assessments. Recognition of common artifacts, such as overfitting or misinterpreted density, and best practices like cross-validation and multiple independent checks before publication. Structure-based drug discovery uses 3D protein models to identify binding sites and design selective molecules, following a process of target validation, molecular docking, and lead optimization, and this approach has been widely adopted to accelerate development of more effective therapeutics. Structural biology reveals disease mechanisms by showing how protein misfolding and aggregation contribute to conditions like neurodegeneration, highlighting specific proteins such as tau, and by mapping side chain changes that link genetic variants to altered molecular function and pathology. Looking ahead, advances in AI and machine learning are accelerating structural analysis by improving prediction, accuracy, and throughput, and future projections suggest that emerging technologies will further reshape the field, enabling faster discoveries and more integrated experimental computational pipelines. This enzyme case study connects three-dimensional structure to catalytic function by using a well-characterized model system to demonstrate how active site architecture determines mechanism, supported by conference insights and additional datasets that strengthen the structure-function link. The receptor-ligand case focuses on the structural basis of signal transduction, showing how ligand binding induces conformational shifts in receptors such as GPCRs or kinases, and explaining why understanding these changes is crucial for designing targeted therapeutics that modulate cellular signaling. Hands-on visualization with Pymal and Chimera teaches practical skills like loading structures, rotating and zooming, coloring by properties, measuring distances, and highlighting secondary features, giving researchers a direct way to explore models and communicate structural insights visually. Structural comparison methods, including three-dimensional superposition and sequence alignment, allow direct comparison of protein shapes and conserved regions, offering essential insights into functional relationships and evolutionary history as new alignment techniques continue to improve. Current limitations include difficulty modeling, dynamic conformations, and transient states that are essential for function, challenges capturing multi-state interactions with static predictors, and prediction gaps that slow full mechanistic understanding despite rapid methodological progress. Ethical considerations center on data sharing and collaboration, where open data accelerates discovery but must be balanced with intellectual property rights. Following institutional guidelines and thoughtful IP strategies helps ensure both broad scientific benefit and incentives for therapeutic innovation. In conclusion, this lecture summarized core concepts and techniques in protein structure analysis, from X-ray crystallography and cryo-EM to computational modeling and AI-driven prediction, and emphasized that the evolving landscape offers exciting opportunities for advanced research, education, and future therapeutic breakthroughs.